Now we will study about biochemistry, genetics, and genetic terminology, all the terms, all the genetic terms, and what are their meanings. First genetic term is codominance. Definition: both alleles are fully expressed in a heterozygous individual. Neither alleles mask the other. So it is the mechanism where both alleles are expressed in the individual, and both allele shows their phenotype, and neither allele masks the other. So both alleles are fully expressed in codominance. They both show dominance and they are equally expressed. Mechanism: gene products from both alleles contribute equally to phenotype. From both alleles, they equally contribute to phenotype. So they both show codominance. For example, ABO blood groups, AB type, where these both are equally, they both allele equally contribute to phenotype. ABO blood group, AB type, HLA groups, alpha 1 antitrypsin variants. Key concept here is phenotype shows combined expression, not blending. Combined expression and no blending. Variable expressivity. Definition: individuals with the same genotype show different severity or features of disease. Mechanism: influenced by modifier genes, environment, epigenetics. For example, neurofibromatosis type 1 or NF1. Its severity differs between patients, so it is example of variable expressivity. So because NF1 or neurofibromatosis type 1 severity is differs between different patients, so it is influenced by modifier genes, environment, and epigenetics. Third medical term of genetics is incomplete penetrance. Definition: not all individuals carrying a disease causing mutation develop the disease. So it is the definition of incomplete penetrance. For example, BRCA1 mutation. BRCA1 mutation causes breast and ovarian cancer, and due to incomplete penetrance, it does not always cause breast or ovarian cancer. So it is example of incomplete penetrance, that not all individuals carrying a disease causing mutation develop the disease. For example, BRCA1 mutation. Fourth genetic term is Pleiotropy. What is Pleiotropy? Its definition is one gene affects multiple organ systems or traits. Pleiotropy is that one gene affects multiple organ systems or traits. Examples of pleiotropy are cystic fibrosis, thick lung mucus, GI involvement, pancreatic insufficiency, male infertility. These are examples of pleiotropy. Its definition is one gene affects multiple organ systems or traits. Fifth genetic term is anticipation. Definition: disease shows earlier onset, increased severity in successive generations. Mechanism is expansion of trinucleotide repeats. For example, Huntington disease. Loss of heterozygosity or LOH. Definition is cancer develops when the normal wild-type allele of tumor suppressor gene is lost after one mutation. Mechanism: it requires two hits. First mutation, that is inherited or acquired, and second allele lost or mutated. In the loss of heterozygosity, important note applies to tumor suppressor genes, not required for oncogenes. Examples: retinoblastoma and Lynch syndrome (HNPCC). So these two are examples of LOH, loss of heterozygosity. Epistasis definition: gene and one locus modifies or masks the expression of another gene. Example: albinism, alopecia. So these two are examples of epistasis, in which one gene at one locus it modifies or it masks the expression of another gene or suppress the expression of another gene. So these are examples of epistasis. One is albinism and second is alopecia. Aneuploidy definition: abnormal number of chromosome. So what is aneuploidy? It is abnormal number of chromosome, cause chromosomal non-disjunction during meiosis and mitosis. So basically there are some, due to non-disjunction of chromosome during meiosis and mitosis, so there are some abnormal number of chromosomes, so it's called aneuploidy. Examples of aneuploidy are Down syndrome, trisomy 21, Turner syndrome, 45XO, and these are seen in oncogenes. So aneuploidy is abnormal number of chromosome due to meiosis and mitosis. So Down syndrome and Turner syndrome are examples of aneuploidy. Genetic terms: dominant negative mutation. Definition: mutated protein interferes with normal protein function. Mechanism is abnormal protein binds DNA or complexes but blocks normal protein activity. For example, mutant p53 tumor suppressor blocking normal p53. Linkage disequilibrium. Definition: certain alleles occur together on the same chromosome more often than expected by chance. Key features measured in populations, not families, varies among ethnic groups. For example, HLA genes. Mosaicism definition: presence of genetically different cell populations in one individuals. So it is mosaicism, presence of genetically different cell populations in one individual. A is somatic mosaicism, mutations occur after fertilization, affects some tissues only. And second is B, germline or good, mutation present only in egg or sperm cells. Parents appear normal but can have affected children. Classic examples are McCune-Albright syndrome, cafe au lait spots, ragged borders, polycytic fibrous dysplasia, endocrinopathies, precocious puberty. Mutation lethal if present in all cells, survivable due to mosaicism. Now we will discuss about locus heterogeneity. So what is locus heterogeneity? Definition of locus heterogeneity: mutations in different genes cause the same disease. For example, albinism, retinitis pigmentosa, familial hypercholesterolemia. So these are examples of allelic heterogeneity. Definition: different mutations within the same gene locus cause the same disease. So it is allelic heterogeneity. For example, beta thalassemia. Heteroplasmy. Definition: presence of normal and mutated mitochondrial DNA in the same cell. So what is heteroplasmy? It is presence of normal and mutated mitochondrial DNA in the same cells. Key features are: it produces variable disease severity, inheritance of mitochondrial DNA passed from mother to all children. For example, Leber hereditary optic neuropathy or LHON. Leber hereditary optic neuropathy is example of this. Another medical term is uniparental disomy or UPD. Definition: child inherits both copies of chromosomes from one parent. Types: heterodisomy, meiosis 1 error. Chromosomes different: isodisomy, meiosis 2 error or duplication. chromosomes identical. Important facts: chromosome numbers remains normal. Euploid, often normal phenotype. Clinical clues are suspect when recessive disease appears, but only one patient is carrier. Examples are Prader-Willi syndrome, Angelman syndrome. Population genetics concepts, bottleneck effect definition, large population drastically reduced, genetic variation decreased. So it is the bottleneck effect, where large population drastically reduced, genetic variation disease decreases. It results rare allele becomes common. Natural selection definition, traits improving survival, reproduction become more frequent. So it is natural selection. Genetic drift definition, random change in allele frequency due to chance events, stronger in small populations. Now we will discuss about summary of this concept and core idea. What is codominance? Codominance, both alleles expressed, variable expressivity, same gene, different severity. Incomplete penetrance, gene present but disease absent. Pleiotropy, one gene, many effects. Anticipation, earlier and worse disease generations. LOH, two hit cancer mechanism. Epistasis, one gene masks another. One gene is dominant and another is recessive. Aneuploidy, abnormal chromosome number. Mosaicism, two genetic cell lines. Heteroplasmy, mixed mitochondrial DNA populations. UPD, both chromosomes from one parent. Now we will discuss about key medical terms used here. Allele, different version of a gene. Genotype, genetic makeup. Phenotype, observable characteristics. Codominance, both gene versions expressed. Penetrance, probability gene shows disease. Expressivity, degree of disease severity. Pleiotropy, one gene affects many systems. Anticipation, disease worsens each generation. Tumor suppressor gene, prevents uncontrolled cell growth. Oncogene, gene promoting cancer growth. Loss of heterozygosity, loss of normal protective gene copy. Epistasis, one gene modifies another. Aneuploidy, wrong chromosome number. Non-distinction, failure of chromosomes to separate. Dominant negative mutation, mutant protein blocks normal protein. Linkage disequilibrium, genes inherited together frequently. Mosaicism, different genetic cell populations. Locus heterogeneity, different genes, same disease. Allelic heterogeneity, different mutations, same disease. Heteroplasmy, mixed mitochondrial DNA. Uniparental disomy, chromosomes from one parent only. Genetic drift, random genetic change. Natural selection, survival based gene selection. Bottleneck effect, reduced population diversity. So these are important terms used in our lecture.